I just don't know if I was just like long pitch and
You're getting like just that.
Test. Yeah. No, I am getting my. Yeah, you should.
I mean, I'm not sure that I would take
But me and
And I'm not going to turn on all
while you're putting it on, so it's not going to put on I'll go, if you want to just test, say 1, 2, 3 or something.
That's working.
How's that go?
I think it's working. Maybe not loud enough. Is it working?
Sounds like he's not hearing it.
Not working? Yeah. So like this? Like this? Try it out. Like this? Yeah, maybe it's good. This is not such a big room, I can hold it.
It'll be fine. It's mostly for the stream.
All right, we're going to get started now. So it's my great pleasure to introduce Nico Kriegeskorte. He is a professor in departments of psychology, neuroscience, and electrical engineering at Columbia University, where he's the director of the Cognitive Imaging Center at the Zuckerman Mind-Brain Behavior Institute. He got started studying psychology, computer science, and cognitive neuroscience at 2 different Max Planck institutes in Germany, focusing on rotation invariant object representation. And he earned his PhD at the University of Maastricht in Netherlands in cognitive neuroscience, focusing on the neuroimaging of the human visual system. He did postdocs at both the University of Minnesota and the NIH, and was a faculty at University of Cambridge for about 8 years, and very recently moved to New York to be at Columbia. It's hard to underestimate how much impact Nico has had on the field of neuroimaging analysis and in vision research. And he does so by marrying both methodological and empirical insights. And so I think this is maybe best illustrated by one of my favorite papers, which is his 2008 Neuron paper, where he took electrophysiology data recorded from monkey IT cells on one hand, and acquired matching human neuroimaging results for the same stimuli on the other, and figured out how to compare them. And in doing so, 2 things happened. First, for the first time, we could just see, with an elegant visualization, just how similar these organizations were. And it started to give us insight into the major organizing structure and the nature of the representational space of these objects in IT cortex. And the second thing was, in doing so, he invented this method, you may have heard of it, representational similarity analyses, that has since then had an impact far beyond vision, and is one of, if not the most dominant methods for understanding condition-rich neuroimaging data, and beyond fMRI even, to date. So currently, he's sort of been at the forefront of exploring advances in computer science, namely machine vision models, and bringing them into neuroscience. So he has a broad vision of the directions that the field can take towards a cognitive, computational, neuroscientific explanation of behavior. And to this end, he co-founded a new conference that's had two meetings so far, the Cognitive Computational Neuroscience Conference. And what he's going to tell us about today, I think, is some of his latest work on how you leverage and modify deep neural networks to gain insights into the mechanisms of biological vision. So let's welcome Nico.
Thank you, Talia, for this too generous introduction. You see, this is a stereoscopic presentation here, so I'd like to ask you to cross views, and you'll see this in 3D, actually. All right, so I'm going to talk a little bit about theory and data in the beginning. So this is going to be very conceptual and somewhat methodological. And then I want to tell 2 stories about research in my lab. That's an example of the interesting things that are happening with neural networks in many different labs at the moment, some of which are here at Harvard, Talia's lab, and at MIT as well. So this is sort of particular perspective on a more general development, I think. So theory and data, let's get into it. We've got these amazing ways of measuring and manipulating the brain at different scales. So for example, 2-photon calcium imaging in small animals and functional MRI at a much coarser resolution in humans. Both of these give us amazingly rich data, brain activity data, as you can see in these examples. But I think we haven't fully learned yet what to do with these data. So when we get these wonderful rich data sets, the first thing we're we're tempted to do is to reduce the dimensionality with techniques like PCA. For example, in this paper, they use PCA to characterize the responses of these large sets of neurons recorded in the zebrafish. And in this paper, similarly, they used PCA to reduce the dimensionality and make these colored maps where we assign selectivities to different neurons. And I think, you know, it's a good first step in terms of getting a sense of the variation, the major components of variation in the data. But we're also all very aware of how far this kind of data analysis is from getting at the computational mechanism, at an understanding of the information processing that's going on in the brain. So how do we connect theory and data? Well, at the level of theory, we can think of different levels of description at which we can formulate models, from the level of single neuron computational models to the box and arrow models of cognitive psychology. And there are these intermediate levels, like biologically detailed neural network models, deep neural network models, which are typically rate-coded models, and cognitive computational models, which are symbolic models of computer science based models of the information processing that are being developed in cognitive science to understand information processing in the brain. And on the side of the data, we have different spatial scales from single electrode neuronal recordings. And of course, there's a lot I'm leaving out here underneath, but we're focusing on this limited range, up to behavioral measures, where we're looking at the organism as a whole and we're observing its behavior and trying to understand what it's doing and what it might be thinking. And in between here, we have neuronal array recordings, high resolution and low resolution imaging. So you could think of fMRI being somewhere in between here, and EEG and MEG maybe more on this side, and calcium imaging would be closer to here in this range. And now we want to think about how we can connect theory to data and how historically different disciplines within the sciences of the brain and mind have attempted to do this. So for example, computational neuroscience started out by linking these computational models of single neuron responses to single electrode neuronal recordings with the goal to predict the response of a neuron to a novel image, for example. And cognitive psychology here, way at the top, has attempted to link these box and arrow models of information processing in the brain to behavioral measures in, for example, reaction time experiments. Cognitive science made this pursuit more rigorous by using computational models that were fully explicit with respect to the information processing, if abstract, and still use these behavioral measures. And cognitive neuroscience attempted to link cognitive psychology to low-resolution brain imaging. So I think when we look at this historically like that, one thing that's obvious is sort of this big hole in the middle here. And that's where the intermediate spatial scales are on the level of the data, where we're bridging the gap between the behavior and what the single neurons are doing. And at the level of models, this is also where the computational models are that occupy this intermediate space where we can begin to think of the information processing in terms of its implementation in the brain. And so the idea is that by putting cognitive science together with computational neuroscience and really trying to meet the combined criteria of success of both of these, we're sort of invading this intermediate space here and linking these explicit brain information processing models to the ever richer brain activity data that we can acquire with modern techniques. So what does it mean to understand how the brain works? To me, it means explaining behavior and explaining neural activity with computational models that are neurobiologically plausible and that perform real-world complex tasks. So on the one hand, we want the model to actually be able to perform the task, and we also want the model to use only components that we can at least imagine being implemented in the brain, although the components might be quite abstract. So abstracting from biological detail is certainly desirable, but it's an open question to what extent we can abstract and not lose aspects of the dynamics that matter to the information processing. And then with these models, we want to explain detailed patterns of behavior, for example, patterns of reaction times across many different stimuli and vision, or detailed movement trajectories, or patterns of errors as well. And we want to be able to explain neuronal activity at some level of abstraction, which is to be defined, right? So this is a very kind of top-down view of what I would expect of a model that for some limited task can claim to explain how task performance is implemented in the brain. And there are lots of sort of wildcards here, things that we have to define, namely exactly what aspects of behavior we want to be able to predict and what aspects of neuronal activity we want to be able to predict. Historically, different disciplines have tackled subsets of these criteria. So cognitive science, computational neuroscience, and artificial intelligence have focused on sort of subsets of these. And cognitive computational neuroscience means to sort of put them all together and try to combine the methods from these three disciplines and meet all of the criteria in the context of some restricted task. So the idea is that with the different tasks that we design, we kind of carve up cognition and we focus on a particular piece of the mind that we can bring into the laboratory and then maybe attempt to meet all of these criteria. And there's sort of different modeling frameworks, as we've seen. There's the cognitive science work that uses these very abstract models and takes more of a top-down approach, and I think that's very important, and that's part of cognitive computational neuroscience. And then there's the more bottom-up approach that's been taken in computational neuroscience, where people start from the biology and then they simulate single neurons, maybe with multiple compartments, with dynamic models and interactions in spatial dendrites. And somewhere in the middle is this recently very successful paradigm of deep neural networks, which are easy to imagine in terms of implementation in a biological brain, but also abstract enough to scale to real-world cognitive tasks. And so neural networks occupy this particular place in this diagram at the intersection of these disciplines, where they've been really very important in the history of each of these disciplines, causing a paradigm shift in cognitive science in the 80s with parallel distributed processing, having been important in computational neuroscience for a very long time, with neural network models at different levels of biological detail being used to understand dynamics within single neurons and in networks of neurons as well. And in artificial intelligence, sort of in the last 6 years, these deep neural networks really becoming a central method that has helped us conquer a lot of different tasks. In vision, they're the only game in town right now, and in other applications, they're at least a component of many systems today, although, of course, probabilistic models continue to be key and can be combined in very interesting ways with neural networks as well. So what is a neural network model? Well, to biologists or neuroscientists, it's almost painfully naive to call these very abstracted model neural networks. That's a biological term, right? And I imagine when the term first came to be used, it was with a kind of naive exuberance that people started calling them that. A more descriptive term would perhaps be differentiable function network models, because they share only the most abstract features with biological brains, namely being composed of units that take lots of inputs and then combine these inputs in some nonlinear way and then are linked up in a network of interactions. So let's talk a little bit about neural network models. So they take these inputs, so we're talking about a particular kind, the one that's usually meant when we talk about deep learning, these abstract models. They take a set of inputs compute a linear combination of the inputs and pass the result through a static nonlinearity, which could be any of a number of different nonlinearities. And then they can be linked up in networks in different configurations. So here there's a shallow feed-forward configuration where everything goes through a single hidden layer. You can have a deep network, meaning a network with more than one hidden layer, where there are these two stages of internal nonlinear transformation. And you can have recurrent networks where these units here are linked up in cycles and give rise to dynamics, and therefore lend themselves naturally to processing dynamic stimuli and to predicting dynamic sequences as well. In these networks, you need a nonlinear activation function to make the hidden layer useful. Imagine you had a neural net with just linear activation functions, and imagine this exact network with two inputs. Every hidden unit here would compute some kind of ramp function over this two-dimensional input. And if you have a bunch of these ramp functions here in the hidden layer, at the output layer, if everything is linear, all you're doing is linearly recombining these linear combinations of the input, so you can never get beyond a linear combination of the inputs, and the outputs will all be just linear functions of the inputs. So as long as you stay with just linear activation functions, you can never benefit in terms of the expressive power of the network from hidden units. So, but if you have nonlinear functions, let's say each of these computes this nonlinear ramp, it's a sigmoidal function over the two-dimensional input. When you combine these linearly, you just add up these ramps, you can make very complicated and interesting functions. And now remembering that by setting these weights here, you can rotate these sigmoid ramp functions around, and you can stretch them, you can make them as steep as you want, and then you can re-weight, adjust the weights here one level up in order to weight them as you're adding them up. This gives you a very rich compositional language for approximating functions. And here, for example, one thing you could do is at the output, select a single location in this input space. And if you think about this for half an hour more, pretty quickly you come to the conclusion that this is a framework that's so flexible that in fact it allows you to approximate any continuous function. So these networks are known as universal function approximators, because given enough hidden units here, you can get arbitrarily close to any continuous function. So this raises the question of why you need deep networks. If even shallow networks have this property of being universal function approximators, so why do you need deep networks ever? Well, the answer is that many deep nets can represent complex functions more concisely. So, or rather, deep nets can represent particular complex functions more concisely using fewer units and weights. So if you're in a realistic domain where you're limited with your units and weights, then the approximation will only be so good. And with a deeper architecture, given the same number of units and weights, you can get closer to the function you'd like to represent. Similarly, you might ask, why do you need recurrent neural networks? Well, recurrent neural networks have the ability to recycle their computational resources through time. So if you imagine a feed-forward network in a limited skull, there's only so many units. So with a feed-forward sweep, you run out of neurons to compute with. But if you can recycle your neuronal resources over time, then you can use time as a resource and compute much more complex things. And also, recurrent neural networks have internal states, so they can dynamically compress the recent stimulus history and deal with a dynamic environment. And it turns out that these recurrent networks are universal approximators of dynamical systems. So this is just reminding us of how general a framework for modeling things these are. And that could be, of course, an advantage, but it could also be a disadvantage. It's a disadvantage in the sense that it's not a theory, but a language in a sense. You can't empirically falsify that something is a recurrent neural network. In order to set all these weights, so we've talked about the expressive power of these networks, but we haven't talked about how to set all these weights. The most widely used method is gradient descent learning using the backpropagation algorithm. So let's just briefly consider this in the context of a deep neural network drawn here. So we have the input. I've drawn 2 input units here, but the inputs might be 1 unit for each of the pixels of an image. And you have the output, which could be a category label. So this could be many of, for example, 100 output units for 100 different categories. And in between here, you have hidden layers with these nonlinear transformations. So gradient descent learning involves, for each training image, adjusting each weight slightly so as to reduce the output error. And the adjustment will be proportional to the error gradient for each weight. And this is equivalent to going down in the steepest direction on the surface of the error when the ground is sort of the space of all weights. So you want to, you can only see locally, and you can sense locally in which direction in the space of weights the error decreases, and then you take a small step in that exact direction. So another way to think about that is that for a given input image, all of these units are in a particular state of activation, and we can linearize this whole nonlinear network about that state. And then it's easy to compute the partial derivatives with respect to each of the weights of the cost function in order to know in which direction to change all the weights. So here's the math for that, where we take a feed-forward pass through the system in order to compute all the activations. And then the efficient way of computing all these partial derivatives is to move backward through the network, because that minimizes the redundancy. There's lots of redundant computation as you compute, for example, the partial derivative for this weight and the partial derivative for this weight. All of the things up here are the same computations, so you don't need to do them redundantly. So the effective way of doing this is to backpropagate the errors. So backpropagation has a long history of debate with respect to its biological plausibility, and there is a new and exciting literature on how it might be implemented in the brain, or how something akin to backpropagation might be implemented in the brain. In my lab, we don't really study this because the target, the thing we want to model, is not the learning process, but it's the perceptual process. So we use backpropagation just as a hack to set the weights for our networks. An important question, if we want to bring deep neural networks into empirical neuroscience, is how we can test a neural network model with brain activity data. So how can we, given a range of neural activity data and a range of models, how can we adjudicate among the models using the brain activity data? So the way we're currently pursuing this is by predicting representational spaces. So we have this idea that in the neural network we have these layers, and the layers contain population code representations, and we want to be able to compare these population code representations to those in biological brains. So how do we do that? Well, we have to predict the structure of the representational space. So let's say we have a bunch of visual stimuli here. We present them to our subjects' brains, and these could be humans or monkeys, and we present them to our models. And then in some brain region and some layer of the deep neural network, we get these activity patterns. So for each particular stimulus, we get a particular activity pattern, and this could be cortex here in 2 dimensions and activity sticking out of the plane. So this is a little sort of pattern, and this will be a different pattern indicating that the region cares about the difference between these 2 stimuli. Now the question is, how do we compare all of these patterns to all of these patterns when we have a a correspondency in terms of the stimuli, but not in terms of the units of the models and the neurons in the brain. So the problem is we have the stimulus response matrix, which we can think of in terms of its columns, the activity profiles or tuning functions. That's the pattern of responses of a single neuron to all of the different stimuli. Or we can think of it in terms of its rows, the activity patterns, which is the responses of all the different neurons or voxels to a single stimulus. And we have such a matrix for the brain region we're trying to understand and for the model layer. And we've presented the same stimuli, so these two matrices match in the vertical dimension, but they might not match in the horizontal dimension, so we don't know the correspondency between these responses, and they might not be the same number of responses even, right? So this is an interesting problem, and in the literature there's 3 different approaches to overcoming this problem. So let's look at this matrix first in terms of its set of activity profiles. We call the stimulus response matrix U, and we can look at the matrix in terms of its activity profiles. So we're looking in a space spanned by the stimuli. So one dimension here is the activity elicited by stimulus 1 in each of the neurons, and one point here is a response, and the coordinates are all of these activities to the different stimuli. So for a number of responses, we get as many dots here as there are response channels, which could be neurons or voxels in fMRI. And we can model this distribution as a linear combination of these basis activity profiles. So these are like basis tuning curves that we linearly combine to predict each of the particular responses that we've measured. And this is called an encoding model. Alternatively, we can look at the same data, this matrix, in terms of its activity patterns. So now we're looking at the space spanned by the responses, and a point in the space corresponds to a stimulus, to the response pattern elicited by a particular stimulus. And we can characterize the geometry of these response patterns by looking at the distances between pairs of response patterns. So that's the distance matrix, and this approach is called representational similarity analysis. There's also a third approach where we look again at the space spanned by the stimuli. So we look again at the same distribution of the activity profiles, but then we model it as a multi-normal distribution characterized by its second moment, the second moment matrix G, and this is called pattern component modeling. It's slightly less known, but it's a very powerful technique with some advantages over both of these, developed by my collaborator Jörn Dietrichsen. So these two look at the distribution of activity profiles, whereas this one looks at the representational distances. And these two use model as a model, the stimulus by stimulus matrix of summary statistics, different summary statistics, the G matrix, which you could think of as a similarity matrix of inner products, and in this case the D matrix, which you can think of as a dissimilarity matrix. And this one models each response separately. So I just wanted to give you kind of a teaser for this paper in which we relate these 3 approaches to this problem of predicting representational spaces and using such predictions to adjudicate between alternative computational models. In this paper, we argued that there is a core commonality to these 3 approaches in that all 3, in their most commonly used implementations, test hypotheses about the second moment of the activity profiles. So I'm not going to explain this completely, but it's sort of an important, I think, step in terms of understanding how these relate to each other. But in the rest of the talk, I'll focus on this technique, which is our sort of standard go-to technique, although we use elements of all of these, and these are all part of kind of the same toolbox of methods that can be mixed and matched as needed. So let's get to the first part of the empirical story, and that's about feed-forward neural networks. And I would call that a converging story. So I think we're going to be building feed-forward neural networks probably for another decade or two, trying to understand exactly what the transformations are across stages of processing and what each of the biological visual areas do, V1, V2, V3, V4, IT, what are their precise roles and differences. So far we haven't modeled that in detail at all, and even for just the feed-forward sweep, I think there's a lot more work to be done there, and that's going to be very interesting. But at the same time, it's a somewhat converging story, I think. So we can kind of see the light at the end of the tunnel, perhaps, of looking at the feed-forward sweep through the ventral stream. And so I want to give kind of a glimpse of this converging story through particular couple of examples from my lab, but this is really a broader development. So first I have to introduce you to a particular kind of neural net, particular kind of deep neural net, called a deep convolutional neural net. So in a deep convolutional neural net, the architecture is defined such that each unit in a layer sees only a little piece of the two-dimensional input image. So it has restricted receptive fields, and that's pre-specified, that's built into the architecture, inspired by biological vision in primates. And then in addition to having these restricted receptive fields, the little weight template through which each unit in the first layer sees the image is automatically generalized across all positions. So there's a unit here that sees this patch of the image through a linear combination of the image with this particular weight template. And there's a unit here which uses the same weight template and applies it to a different patch of the image. And that's happening all over a so-called feature map for a given feature like this. And the features are learned by backpropagation, but this fact that they're the same, called the weight sharing, is pre-specified. And now you learn a bunch of these feature templates, and this computing a feature like that for all locations amounts to a convolution of the image with that feature template. So that's why they're called convolutional neural nets. And the outputs are passed through a static nonlinearity, often this rectified linear activation function. And this is often followed by some kind of local pooling where we decrease the spatial complexity of these outputs, for example, taking local maxes or averages. And often this is also followed by local normalization. All of this is called a single layer, and these layers are stacked with the output of this layer forming the input of the next layer. And over stages, we progressively reduce the spatial complexity of the representation while increasing the feature complexity. So we're going to take an approach to testing these kinds of models based on representational similarity analysis, where we look at each pair of stimuli here and compute a measure of the similarity. We use cross-validated Mahalanobis distances in order to estimate the discriminability for each pair of stimuli in order to make a matrix where for each pair of stimuli we can look up how discriminable these two stimuli are in the code. We call this matrix the representational dissimilarity matrix, or RDM, and we're going to be comparing model representations to brain representations by comparing these RDMs, typically using a correlation coefficient. In order to do this comparison, we often want to be fitting some parameters. For example, we might assume that we have a model that has the right set of features, but we might not know exactly in what proportions the different neurons represent these different features. So we want to have some flexibility, and there are different ways of doing this. One is to allow general linear remixtures of the original feature space. Another is to just allow the feature space to be stretched or squeezed along its original axes to make for a somewhat more flexible fit. In either case, we need to train the model on our brain activity data, and where we do such fitting or training, there is overfitting necessarily. So we want to train and test in cross-validation. So I want to just briefly show you how we approach this at the moment. So we have these RDMs, these matrices of representational dissimilarities, and we choose a set of training stimuli, and that corresponds to a subset of these dissimilarities. And then we have repeated measurements in different subjects, and these are our training subjects. And then we consider a different set of subjects, our test subjects, and in those test subjects we consider a disjoint set of test stimuli, such that we can fit and test on two different sets of training and test dissimilarities where there's no overlap in terms of either the stimuli or the subject. So this is important to not overfit to either the stimuli or the subjects, because like in computer vision, a computer vision model doesn't work if it doesn't work on new images. And so similarly, if our ambition is to have a model of the ventral stream, if it doesn't work on new images and on new subjects, then it's not really a valid model that engages the complexities of that system in the brain. And then around this cross-validation, which enables us to compare how well different models perform at predicting the representational dissimilarity matrix, we wrap bootstrapping of both the stimuli and the subjects, because we want to treat both stimuli and subjects as random samples from a population of stimuli and from as a sample from a population of subjects. So this is our current approach to doing the inference. So here I'm going to show you results for a particular network, and that's the famous network called AlexNet, which in 2012 convinced everyone in computer vision that neural networks are the way to go. We're doing this for a range of different networks. I'm going to show you some slightly newer and deeper networks in a moment. But overall, what I'm going to show you here is pretty much still the state we're at, even with these more complex models, in terms of predicting representational geometries in IT. So on the vertical axis here, you see the accuracy of the human IT dissimilarity matrix prediction, and that's the group average of the Spearman's row rank correlation. And as we go up layers here, for layer 1, we get a very small correlation. And as we go up the layers, the correlation with the human IT dissimilarity matrix gets higher and higher. Here we're in the fully connected layers. And we get the best result when we use a weighted combination of the layers and a set of SVM readout discriminants. So this is fitted with this cross-validation method. So most of these are significantly related to the representational distance matrix for human IT, and that's a very low bar to go by. But all of them are below what we call the noise ceiling, which is an estimate of the range in which the true data generating process would fall if it was one of our models, if that makes sense. And we have an upper bound and a lower bound on this noise ceiling. The upper bound is a hard upper bound. It's the highest accuracy that any model can achieve based on the fact that the single subject IT RDMs are sort of a bunch of RDMs in RDM space. And what I'm plotting there is the average correlation. So there is no RDM that is perfectly correlated to all of them on average, right? So there's some RDM sort of in the middle of this cloud that maximizes the average correlation, and that correlation would be this one, right? And then there's the lower bound of the noise ceiling, which is the other subjects average as a model for each of the subjects RDM. So you can think of this one as an overfitted, so this one is actually obtained by taking all of the subjects average and then correlating it with each of the subjects and computing the average. That happens to be the one that maximizes this for Spearman correlation. And you can also do this in cross-validation, and then you get a lower bound on the performance of the true model, because your stand-in for the true model is the average of the other subjects, and that's also corrupted by noise, so you expect that to be negatively biased as an estimate of the performance of the true model. So you have this range here. And then we can do all the model comparisons as well, and we find that this weighted combination here outperforms most of these other models. Here for comparison is the performance range of 27 different computer vision features, which are these hand-engineered features that we've been working with for a couple of years before starting to use neural networks. So these models are clearly better than computer vision features, and there's clearly a big step forward in terms of this works similarly well on monkey IT cell recordings that we also worked with in the same paper. We analyzed these monkey IT data as well. You can also do this with encoding models. People in Jim DiCarlo's group have done a lot along these lines. And in general, what this has meant for the field is that we're now in a position to predict responses of neurons to novel images in high-level visual areas like IT. So this was always the difficult challenge of visual computational neuroscience, and there's been some real progress there, so that's wonderful. But it's important to remember that, of course, we're not explaining all of the variance. There's still a gap here to the noise ceiling, and these are experiments that focus heavily on static stimuli and brief presentations, so they kind of emphasize what's going on in the feed-forward sweep of processing through the ventral stream. And that is a small part of what vision is about, because vision is a much richer process of dynamically engaging with the world around you and drawing very deep inferences about all the things and how they relate to each other and how they relate to your goals and to the future. Sure.
How much is this really depend on the way you train the network? So it's technologically it's amazing that these convolutional networks can perform classification maybe beyond human level. But that can guarantees that the representation to perform that task is obtained, regardless of how it's obtained, whether it's biologically similar. So the fact that this network performs this particular task that you train kind of guarantees that the representation is prepared to perform this task. And that is kind of mimicking how humans perceive these stimuli.
Yeah, that's a deep and important point. And there's a number of things in my talk that partially address that, but actually not fully. So I would love to discuss it with you in more detail. But here's there's a few points coming up that kind of get at that a little bit. So one thing is that what we're plotting here is not the performance of the model at the task of categorization, but the accuracy of predicting this representational geometry. And there's many representational geometries that would lend themselves to classification. So it's not a foregone conclusion that this would happen. But there's also, you see in a moment. So there's this. Deep net layers correspond to stages of the ventral visual stream. So, for example, this network has been trained on classification, but its early layers do very well at explaining early visual area representational geometries. And that's not directly constrained by the task training. And then here in a higher area of lateral occipital complex, it's the later layers that best explain the representation. Here's work from Marcel van Gerwen's lab. So particular example chosen among a number of papers that have mapped out how well different layers of deep neural networks explain different patches of the visual system. And here you see color-coded in blue the patches of the system that are best explained by the low levels of a deep neural network, and in warm colors the patches that are best explained by the higher layers. And you see this continuous progression, so that's another example of something that is not a foregone conclusion, let's put it that way, based on the task training. We can also ask, and again that also goes in the direction of addressing that question, whether the architecture matters. There's a question there.
I just had a small question in terms of the layer questions for organizing it all by layer, but I guess in principle you could just ask for sort of like an arbitrary linear combination of the layers, and by cross-validation you could possibly, you know, find, I mean, do you find that actually sort of single layers best explain a given area? Do you sort of allow yourself to go
yeah, that's a good question. So we're exploring all of these ways of doing it, and linear combinations of layers often work well. Actually, what I showed you, the final bar, was a linear combination of different layers. But you pay a price for fitting too many parameters, right? So there's some interesting questions there in terms of what prior you want to put on the representational hypothesis space, if you will. Right? You can play this game in many ways. At one end of the spectrum, you just say, let's take this layer and let's not fit any parameters to the neural data. And that's attractive in that you're making a very specific prediction, and it's just based on optimizing for a task. So that's also conceptually nice because it supports it addresses the why question to some extent, right? It would argue in favor of the system having been optimized for that task. But you can also allow some degrees of freedom, or even a large number of degrees of freedom, and then learn from your brain data. And that would be closer to the classical approach in computational visual neuroscience, where you do nonlinear systems identification just based on the brain data, right? And these are interesting discussions where we have to find a good balance. So an interesting question is whether the model architecture matters. And again, that gets at this question of, is it just the task training that explains these results? So here I've plotted the network layer, and here now there's many different models, including these deeper models like this 50-layer deep ResNet. And we're comparing the internal representations across all the layers, and you see that now there are many points here because there are many intervening representational stages to the human IT representational space. And what you see here's good old AlexNet, and none of them really reach the noise ceiling in this example. Many of them have kind of, they have more complex trajectories. They go up and down across these layers. And the ones that do best at performing the task, here's classification performance, where the deep ResNet and GoogleNet is actually the highest in this set. They are not the ones that best explain the brain representation. Again, here this is to be taken with a grain of salt because of the issue that came up in the question before. This is just treating each layer as a precise prediction of the representational geometry. What would happen if you allowed to fit aspects of this, to reweight the feature maps or reweight individual units? That's a different question, right? And so we're in the process of figuring this out. So another question is whether the model's visual diet matters. By the way, how am I doing in time? All right, I got to speed up a little bit. So we asked whether deep nets with domain-specific training explain representations in category-selective regions. So here we used a single architecture, the VGG architecture, and then trained either with objects and animal images or with face images or with place images. And we see that when we train with face images, then in the higher layers, we best explain the fusiform face area. And when we train with place images, we come very close to the noise ceiling here in the parahippocampal place area. So the actual visual diet, the experience given to these networks matters a lot. And if you just take random weights, which are also computationally interesting because they allow you to do feature expansion and do some kinds of classification surprisingly well, you don't really do well at explaining these brain representational spaces at all. We can visualize the internal representations learned by these networks by using backpropagation and then going through the weights and the activations down to the image layer, and then using gradient descent to optimize the image without learning anything in the network, so as to accentuate the pattern that an initial noise image elicited in some layer in the network, for example, hidden layer 2 here. So when we do this, I'm going to show you this in parallel for the VGG architecture trained on either objects and animals or on faces. And we're going to go through the layers. So in layer 4, you see sort of some structure emerge that looks qualitatively similar between these two networks. And as we ascend the layers, we get more and more complex structure, and at some level kind of recognizable elements of natural images to emerge here. So in layer 12, it becomes quite highly meaningful, and then in layer 14, you can clearly see sort of features of animals here. In layer 15 here, you have something like a super face with lots of features of faces kind of psychedelically combined to strongly drive this highest layer in this network that's learned to identify faces.
So Nico,
Yeah.
if you fed these pictures back to the network, it would classify them with high confidence that these are faces or animals?
Yes.
And you think that if you gave
I-
it to people, they would?
Actually, I don't have that data, but I would expect that,
Yeah.
yes.
I mean, wouldn't you say that, I would interpret this as in some sense an conceptual argument against these models as models, for example, face recognition, because it would suggest that like we actually have stronger kind of structural constraints that would militate against this kind of like psychedelic face representation.
Yeah, that's interesting. I mean, we actually, this is not optimized to drive the network to consider it a face, but it's optimized to elicit a highly modulated pattern in layer 15, right? And if you could do that for a human, who knows what this would look like? Perhaps it would look exactly like this. Probably not, right? But it's interesting to use this to close the loop to make very bold predictions, ongoing work, making bold predictions from these networks, such as that this is a very strong stimulus for certain higher level representations. That's interesting to test. So the converging feed-forward story is that deep convolutional feed-forward neural networks go a long way toward explaining how the initial sweep through the primate visual hierarchy enables recognition at a glance. These networks predict representations of novel images better than any alternative current model. Both the architecture of the model and the task training contribute substantially to these successes. However, we need to build models whose architecture more closely resembles the visual hierarchy. And a major feature of biological neural networks is recurrent signal flow. So this brings me to the final part of the presentation, where we're going to talk about the emergent story on recurrent neural network models. So we want to start from this model, which I think of as the new null model of ventral stream processing, which is a feed-forward convolutional neural network. So we want to change one thing at a time, right? And therefore, our first step in that direction is just to make these convolutional neural networks recurrent. So what do I mean by that? Here you see layers of a network, and in a bottom-up feed-forward convolutional neural network, each unit just sees this restricted receptive field in the layer below. So we're exploring these different architectures where we add lateral connections. So we have bottom-up and lateral, and we call that a recurrent convolutional neural network, where each unit now sees not only the restricted receptive field from the layer below, but also a restricted receptive field of its neighbors in the same layer across all the feature maps. And then we have the BLT architecture. In addition to the lateral connections, we have these top-down connections. So now each unit also sees a restricted receptive field in the layer above. And since these connections are forming cycles now, this is a recurrent neural network, and it needs to be trained with backpropagation through time, and it generates dynamics. However, it's still convolutional, so these lateral and top-down weight templates are still automatically generalized across spatial positions, and this means that we don't need to learn sort of a crazy number of parameters, and it's still a tractable framework for training on images. We tested this first on this little toy task, which was designed to be hard for feed-forward convolutional networks, where we showed these images. Here's a target 3 in the background, and it's occluded by this, what we call digit debris. So there are all these pieces of other digits on top of it, and the motivation was that this we thought was going to be hard for feed-forward networks that identify these little pieces of things and then pool the evidence with linear combinations one level up. So we saw, for this light level of debris, that our recurrent models, BT, so bottom-up and top-down, BL, bottom-up and lateral, and BLT, with all of them together, outperformed, so had smaller classification error than our feed-forward models. And as we made the debris heavier and heavier, this advantage actually became stronger and stronger. And here are all the model comparisons, most of which are significant. So this suggested that this intuition, which has been in the literature for a long time, that recurrent processing might help deal with occlusion and with more complicated noise signals, seems to be correct. We also tested it on a second task that we call digit clutter, where you have multiple digits, and you're supposed to just say all the digits that you see there. And there too, the recurrent versions of the network significantly outperformed the feed-forward models. Where this is sort of the naive feed-forward model with the same architecture as these recurrent models, but only the B connections. And these two are parameter-matched, approximately parameter-matched control models. An important question is whether recurrent convolutional networks can be scaled up to natural images. So in a current study, Courtney Spura, who was also the lead author of this project with digit clutter and digit debris, is scaling it up to ImageNet and to a new image set that we call EcoSet, which has slightly more ecologically valid balance of different categories. And he's using a range of different architectures. These are all feed-forward, with these being parameter matched to our recurrent models in different ways, by broadening the features, by adding more feature maps, or by adding more layers so that the depth is actually twice the depth of the recurrent convolutional model. And what we see is that the recurrent models seem to outperform these feed-forward control models, including these models that have the same number of parameters, approximately, but in different architectures. And interestingly, these recurrent models can trade off accuracy for speed. So here what I'm plotting is the recurrent computational cost. As you keep running the network, the accuracy rises, and you can stop computing at some entropy threshold. So the output is a probability distribution over all the classes. You can compute the entropy of that. That's a measure of the uncertainty. And you can say, I stop computing when I've reached a certain entropy. So we start at high entropy, and then we get more and more uncertain. We can put a cutoff here. And in this way, the network can naturally trade off the computational cost for a particular image against the accuracy. And what we find is that this speed accuracy trading recurrent convolutional network outperforms these feed-forward control models. And when we stop it earlier, where its computational cost equals that of these feed-forward models, then it performs about as well as these feed-forward models. So that's kind of an interesting feature of these models. Another question is whether neural network models can capture the representational dynamics in the human ventral stream, and this is my the last bit of this presentation. So we're looking at these representational dynamics for these particular stimuli here, and we're looking at different brain regions, and we're looking at sort of 3 clusters of regions: early visual regions, intermediate visual regions, including V4 and LO, and high-level ventral stream regions, IT and PHC. We're looking at this with human magnetoencephalography, where we do source localization first, source projection using the minimum norm, and then we get these spatiotemporal response patterns for each of these regions. So in each of these regions, we have multiple channels, and we have temporal detail in response to each stimulus. So if we do this in a representational similarity framework, we get for each of the regions the RDM as a movie, right? We can look at the discriminability for each pair of stimuli over time. And so I'm going to show you these movies here for the early visual, the mid-level, and the high-level ventral stream regions. So here the dissimilarity is color-coded with yellow indicating high dissimilarity. And time is going to start at 0, and I'm going to slow this down 100 times, 100-fold, as we watch these movies. In the beginning, this is frame 0, so you see only a noisy pattern here, since all of these are sort of relative to each frame, so they're always scaled to fill the entire scale. So we're going to watch these RDM movies here, and underneath you're going to see the MDS color-coded for categories in this row, and here you see the actual images and how distinguishable they are. So you see when you watch this that after maybe about 60 milliseconds, these categorical divisions come up here, a strong cluster corresponding to human faces. A little bit later, all the faces together, human and animal faces, and other categorical divisions sort of come in and sometimes disappear again. It's a very complex dynamic picture and an interesting challenge for us to figure out what's going on there. So I'm going to show you a couple of more bottom-up inspired analyses of this data set that suggest to us that considering this a purely feed-forward system, or even a slightly modified feed-forward system, is a mistake and is missing major components of what's going on there. So here, first I'm going to show you as a function of time how well the GIST model, which is a low-level feature model based on Gabor filters and combinations of Gabor filters, does at explaining the representational geometry for each of these regions as a function of time. So here we see that early on, so after like 70 to 110 milliseconds, this GIST model explains a lot of variance in all of these regions actually, especially in the early region. And then between 100 and 200 milliseconds, this falls off almost to baseline for these higher level regions, but it stays high for the early regions. So these GIST-like geometries first emerge in the early visual areas where they remain strong. You can also look at the categorical clustering here for faces, and we find that this peaks at a little bit after 100 milliseconds, strongly in all three regions actually, and most strongly in IT, which contains the fusiform phase area. But it's also interesting to note that while the stimulus is still on, this almost entirely vanishes, consistent with a highly dynamic process in the ventral stream. If we look at animacy, we see a very different picture, and also sort of, to us, intriguing and mysterious picture, where between 100 and 200 milliseconds, we see the strong peak of animacy in IT, and with much weaker animacy clustering in the other two regions of interest. Then this almost completely disappears, and then reappears most strongly in V4, as though this division is sort of brought back or fed back, and then emerges strongly in V4. So it's very intriguing kind of dynamics that we are trying to make sense of at the moment, but don't really have a good explanation for. We also looked at Granger causality between frames of these RDM movies, and we see that when we look at the relationship between the early region and the mid-level region, the feed-forward influence rises very early, and the influence in the opposite direction rises a little bit more slowly, but there's significant evidence for Granger causality in both directions. And if we look at V4LO and IT, we see that the feed-forward influence again rises very early, and the feedback influence comes up and then becomes very strong much later in time, maybe about 140 milliseconds. Finally, we're comparing the feed-forward and the recurrent models directly by seeing how well each of these models can predict the representational dynamics. And we test these two architectures. One is a ramping feed-forward architecture, which is our version of a feed-forward model, where we've allowed it some inertia so that the feed-forward model can predict dynamics at all, because we think that's a more conservative control model. And then we have this fully recurrent BLT convolutional model. So we train these models by constraining them directly to reproduce these RDM movies using a technique called representational distance learning that a PhD student, Patrick McClure, who's now postdoc at NIH, developed in the lab. So here when we look at these movies predicted by V4, this is the data, and this is the BLT layer 4, we see this complex dynamics in BLT layer 4 that's quite a good match. I'm going to show this again to the V4LO layer here, whereas the B model, which is this ramping feed-forward model, which just has these recurrent connections here that are constant and therefore can just sort of ramp up its activity, is dynamic only for a moment and then converges to its final representation quite quickly, whereas this one sort of reproduces in a much more detailed fashion the dynamics. So when we compare these models in terms of how well they do at predicting the dynamics, we see that the BLT recurrent convolutional models in all three regions do significantly better at predicting the magnetoencephalography data as well as the functional magnetic resonance data. So in conclusion, the emerging recurrent story looks like this to me. Recurrent neural networks provide a neurobiologically realistic and computationally powerful modeling framework. Recurrent processing can enable a network to use time to recycle its limited computational resources, to trade off speed and accuracy, and to perform the inferences more robustly. Recurrent models also better explain the representational dynamics of the Schumann ventral stream. These, of course, are just first steps, and future work should explore the much richer dynamic repertoire of biological neurons and discover the computational functions. So the general conclusions are that neural networks give us a language for modeling that connects the cognitive level to the biological implementation. The current neural networks that we have fail to perform at human levels on many interesting tasks, right? So neural network AI is very far from human level in general. And these models also only use a fraction of the dynamical components of biological brains. So this, I think, bodes well for taking further inspiration from the brain, which has these amazing capabilities that we can't reproduce yet in engineering. And in my lab, we're taking a divergent approach where we're inspired by the biology but guided by engineering and add neurobiological features to our models to understand their functional contribution. And these include more realistic architectures, more realistic training, recurrent connections, and also stochasticity for representing uncertainty and other functions. And we take a convergent approach by using brain activity to adjudicate statistically among sets of recurrent deep neural network models. Thank you very much.
Great talk, thanks. I was wondering whether you had thought about trying to use this dynamic the recurrent models to introduce you about human behavioral data for object recognition. Like if you ask people to exhibit responses, could you then use people's choices and response times to fit the time constants for the different dynamics, speed forward, recurrent, lateral, and so on? And one reason to do that is because then you could, once you had if you could fit the time constants, then you could go back to the neural data and have a more precise, like a sort of a, unbiased prediction about what the dynamics of the neural data should be.
Yeah, I think that's a great idea, and there are many ways to play this game, right? But the one that you've outlined is an attractive one because you would use the behavioral data as a constraint for fitting parameters of models, and then you'd be closer to the why question in a way, right? We have played this in a different way by just training on the task and then fitting readout filters and seeing across all the images how far from the decision boundary different images are. And we're doing very well at explaining reaction times based on that. So things that are far from the neural network's decision boundary are easy to recognize for humans on average, and that's quite a strong effect. But taking that to the dynamic regime where you're modeling the evidence accumulation in a dynamic neural network is something we haven't done yet. And it's interesting to play it both ways, right? As you suggested, use the behavioral data as a constraint to train the models, but also to train the models, maybe recurrent convolutional models, only on the task, and then predict in a detailed way the behavioral data.
The problem is just that you don't know the mapping of the time constants like the network is in this abstract temporal space, right? And you want to map it onto to the time constants of human brain computations, right? So I don't know how to do that by just training on the task.
Yeah, so I mean, with the recurrent convolutional models, you have a dynamic output, right? Yeah. So you can, for example, impose an entropy threshold and you're predicting reaction times, right?
Yeah.
And so maybe you need a scaling factor, what is 1 second in the world of the network, but you can predict distributions of reaction times with these.
So that's assuming that everything, all time is rescaled, right? But what I'm suggesting is that it could be that like the lateral time constant, lateral recurrent time constant is different from the feedback time constant. You have to fit those separately.
That's also fascinating, yeah.
Something that wasn't clear to me. So there are two approaches. You can teach a network to solve some cognitive task that a human solves, and then check whether the neural activity in the network is similar in some way to measurements from the humans. And the other approach is you can train a network to simulate the activities that you see in a human, and then maybe also check if it produces some task stability or something. So in your talk, it was mostly the second version of fitting the activity of model neural networks to activities in human brains?
Not really. I would say it was mostly fitting the networks on task constraints and then testing them in terms of their predictions of brain activity.
So what is the test for the network in your
categorization?
And the humans were also doing categorization tests, or just showing images?
That's a good question. So in the first, in both the fMRI and the MEG experiments, the humans were doing an irrelevant task at fixation. So color discrimination in one of them. So they're passively viewing the images.
So it's a little different, the task that
Yeah.
the network is solving in the humans?
Absolutely. So the motivation, so all of these are interesting combinations whose interpretation is radically different, and there are different ways of playing it that are interesting. But the justification for why it's okay to do it the way we did it was that the task training is sort of a stand-in for your experience, where you're learning to recognize all kinds of things, but then this knowledge and this ability is ingrained in the weights of your neural network, and that will automatically manifest itself when you passively view an image.
So one big difference between deep learning network and actual brain is the number of layers.
Yeah.
And what makes the real brain perform so well with fewer layers? And one possibility could be recurrent connections. And
I think so.
I see recurrent.
Yeah. So you can achieve the same logical depth and number of nonlinearities by using time as a resource and computing recurrently. Of course, it's not the same thing because you're recycling the same transformations, right? So you're learning different transformations, and there's something very deep there. So that could be a bad thing because it limits you in terms of your number of parameters and expressive power potentially. But that could also be a good thing because it regularizes the model, and it will learn a different way of computing the desired function. Good to see you again. It's been a while.
Yeah, we have to run to our Vision Lab meeting right now, actually.
Sure.
But